Hi everyone, I'm Jillian, and in this tutorial I'm going to walk us through how I used the UCSC genome browser to explore whether DNA methylation at the TP53 promoter corresponds to differences in TP53 expression across two different human cell types. So the UCSC Genome Browser is a powerful visualization tool that integrates large-scale genomic datasets, and what makes it stand out is its ability to overlay multiple types of data all in one view. In this tutorial, we'll look at the ENCODE project methylation and RNA-seq data, and apply it directly in UCSC, comparing the two cell lines GM12878 and the HeLa S3 cell line. And I chose the GM12878 cell line because it is a human lymphoblastoid cell line, and HeLa S3 is a cervical carcinoma cell line. And they have been widely used in TP53 studies to demonstrate cases where abnormalities or mutations in those cell lines have led to cancer development in these tissues. So let's go ahead and get started with the tutorial. Um, so what we're going to do, just a brief breakdown, we're going to locate the TP53 gene in the UCSC genome browser. We're going to add CPG island annotations as well as adding data from the ENCODE project through whole genome shotgun bisulfite sequencing methylation tracks as well as RNA-seq tracks. We'll locate the promoter region of the gene and analyze and interpret some differences between the two cell lines. So I'm going to go ahead and open the browser so we can just go ahead and type in genome.ucsc.edu. This is the main page where it'll bring us. I'm going to click on Genome Browser here. Make sure that you have human selected here. It says human assembly right here, so we know we have the right species. I'm going to select this one, the December 2013 assembly, and then click Go. So this is just kind of what we see. We have a whole bunch of different filters that are automatically set to either hide or these ones up here are dense, pack, show. Those are the few different options. Um, I'm going to right here type in TP53 because that's our gene of interest and select this one. It'll refresh and show us the gene along with some variations of that gene. Um, and just by looking at this, TP53 is on the minus strand, so the transcription goes right to left. You'll note this because these little arrows here are pointing from right to left, indicating it's being transcribed from the 3' end to the 5' end. And because of this, the promoter will be located on the right side of the gene model since that's where transcription begins. So I'm going to zoom in a little until I can clearly see that first exon, which is the thick blue box. You'll notice that when I hover my mouse over one of these thin blue lines here, that is the presence of an intron, and it'll even tell you which one out of how many total there are in that gene. So I'm going to just keep swiping over. I'm going to zoom in once more. So you can see here, this is that first exon. So the transcription start site is around this area with the promoter region being over here, which is where we want to focus for the rest of this tutorial. Alrighty, so now I'll show you how to add some basic annotations from the tracks that are readily available to us. I'll use the visualization of CPG islands to show how methylation patterns can be predicted using UCSC. And I chose CPG islands because they're usually located near the promoter region of a gene, which is this area, either a little bit, probably a little bit upstream of it, which is over here. Um, and they are typically unmethylated in actively expressed genes. So I'm gonna scroll down. So these are all of our different track settings. You'll notice that you can click on each one and it'll define it for you. So if you're um, still a beginner using this genome browser, you can simply just click on each one. If you're not sure what it is, it'll give you a description, and sometimes there's even other filters that you can set within that individual annotation. I'm going to scroll down to the regulation track and I'm going to set the CPG islands to show and then click refresh. And now when I scroll down, it's going to be right here. I'm going to just click on it here so it'll expand the view. So notice we don't see any in frame right now. I'm going to just slide a little bit this way. And we should start seeing some pop up. I'm going to just zoom out a bit. Okay, there we go. All right, so we have one over here upstream of the transcription start site. You'll notice that the darker green the band is, the more methylation is occurring at that CPG island. So I'm gonna go this way too, just to see if there's any others. And yep, we have one right here as well. Um, this one is indicating a lower expression level of methylation as compared to the other one, since this one is a lighter green. Um, Another filter I think is useful under genes and gene predictions is RefSeq. So I'm going to set this to full. Uh, this will display any protein and non-protein coding genes that are collected from the NCBI RefSeq gene bank. I'm actually also going to set this GenCode version 49 to full, which also displays any protein coding and non-coding genes that are mapped against any annotations to our reference chromosomes. So I'm going to refresh that. And here we have a whole bunch of different variations of different references of TP53. This just kind of confirms its overall structure and location for the promoter region and transcription start site. So like this is the one that we initially started off with, but you can see there's a whole bunch of other ones from GenCode and from NCBI. These are all different variants of TP53. So that just kind of confirms the structure and positioning for this gene. I'm gonna hide those for now, just so it doesn't get too overwhelming. And then next, I want to use the ENCODE project to pull readily available information on both DNA methylation and RNA expression in two different cell lines to better understand how these methylation patterns influence the overall expression in TP53. So I'll first start with displaying DNA methylation patterns. To do this, we need to add our own track hub. So to do that, we're going to go to My Data and I'm going to select track hubs. And then I'll want to find the encode DNA track hub, which is right here. I'm going to click connect. And it'll ask me to specify the specific assembly since we're doing this December 13th one. That's the one I'll click on. And then you'll notice it'll add that hub, that track hub down here with a whole bunch of new filters. And so I'm going to use whole genome shotgun bisulfite sequencing for my two chosen cell lines. I'm going to click on it. And my two chosen cell lines were this GM12878 and this HeLa S3. So I'm going to ensure that I have the methylation state CpG replicate 1 selected for both of these. So I'm going to go ahead and do that. Um, I didn't choose to select the second replicate just to ensure that I don't get confused with the data. But you can always go back and select more data if that's desired. And we're going to scroll down just to make sure that those are the only ones selected down here. I'm going to set them to full and then submit. And then you'll notice at first it's pretty zoomed out still, so it'll be a little overwhelming. But this is what you'll see at first glance. Again, what I'm going to do is I'm going to focus in on that transcription start site on TP53 right here. So I'm going to just go ahead and zoom in a bit and try to get that centered in frame. All right, so here we have the TP53 transcription start site in this area. And you'll note just from first glance, there is a bit more activity in this area in both of these cell lines than there are elsewhere. And this can give some indication as to the methylation patterns and how expression will be affected due to an increased methylation near this transcription start site. To confirm that, I'll want to add the RNA-seq tracks to observe RNA expression. So to do that again, I'm going to add my own track from ENCODE. So I'll go to my data again and then track hubs. And then I'll want to find the ENCODE project hub that states RNA track hub, this one right here. I'm going to connect again, ensuring I'm clicking on this one here. All right. And then you'll note that it went ahead and added that one right below that previous one. Um, and here I'm going to click on this RNA-seq by biosample. That will allow me to choose from hundreds of different samples to observe the RNA-seq expression of them. So I'm going to go ahead and locate the two that I am focusing on. So here's this first one. I'm going to click that. I'm going to just click on it, and for this cell line, I will choose the samples that match the ENCODE ID from which I took my original data. So in this case, it was this one right here that ends in COQ. I'm gonna just do the first replicate for both the minus and plus strand, just to make it a little more visually pleasing and not overwhelming. So I'm going to go ahead and deselect all of these ones, and then I will select this one. And those top two are the ones that I want. So I'll just unselect all of these. I'm going to make sure these say full here. Confirm that it's the poly A track. Let's see. And then I will click submit. And now we should see two different poly A RNA-seq signals, both from the forward and reverse strand of this first cell line. And another thing is you'll notice that when I move my mouse across these peaks, the higher peaks indicate higher transcription, and that can be indicated by this decimal point that is changing when I move my mouse. Now I'm going to go ahead and do the same thing for the HeLa S3 cell line. I'm going to find it. I'm going to click on it here. And again, I'll do the same thing. So selecting the first replicate for the minus and plus strand. For the encode ID, the one that I chose was the CPR. So I'm going to just make sure that that is, again, the only one that I have here, just to make things less confusing. All right, and I'll ensure these are both set to full and submit. All right, and here they are. They should be added right up against the previous two that we have here. And you can note now that on both reverse strands, this one and this one here for both cell lines, they both have a relatively high level of transcription within the transcription start site region of TP53. And so this visual comparison already suggests a relationship between methylation and expression. Um, and there are some optional steps that you can do to just kind of confirm the relationship between methylation and expression. But I find that adding extra tracks helps to further the evidence to support the big picture question here. Under the regulation track, I'm gonna find that. Um, I'm going to locate this encode regulation and click on it. I'm going to select DNase hypersensitivity. I'm going to click on that. I'm going to click on that. And I'm going to just make sure that I am again selecting the cell lines that I have been using this whole time. I'm going to just go ahead and deselect all of these. All right. I'm going to set these to full and then go ahead and submit. And then you'll note those will show up right here. So we'll have the DNase hypersensitivity for both the lymphoblastoid cell line as well as the cervical carcinoma cell line here. Um, what this does is it will show any open chromatin that's accessible to transcription. So um, the darker this band is, the more open the chromatin is. Um, and I'm gonna just do the same thing for the other one. So that was DNase hypersensitivity. I'm gonna just deselect this one that's automatically selected, and I'm gonna select this H3K4ME3. Um, what this one does is it marks any active promoters, so that should be pretty consistent as far as location in which methylation occurs. So I'm going to click on this. And unfortunately, the HeLa S3 cell line is not listed here as one of the available subtracks. So for now, I'll just go ahead and click this one. It really just depends on what kind of data is available. But for now, we can still display this one. I'm going to make sure this is set to full and then submit. And we should be able to see that one as well. All right, yeah, so that'll be right here. So you'll notice again that it kind of peaks up a bit, right where this first exon is in the transcription start site. The GM12878 usually shows a stronger H3K4 methylation peak and hypersensitivity, which supports active transcription, again, due to this darker band here. Whereas the HeLa S3 cell line didn't show as strong marks here due to the lighter shade of gray. And this can support the idea that promoter silencing is slightly stronger in the HeLa S3 cell line. And now that you have all of this data, there's multiple ways that you can extract this data into a different format. So I will show you if you want to get some quantitative values from what you've downloaded here on UCSC. I will show you one way to do that. So we can go to Tools up here, and then I'm going to click on Table Browser. And then I'm gonna just ensure that I have the correct group chosen. So you do unfortunately have to do this separately for each hub that you chose, unless you want to do all, but in this case, we focused primarily on methylation and RNA-seq. So I'll just show you with the ENCODE DNA track hub first. All right. And then I'm gonna scroll all the way down. So these are the methods, and we looked at whole genome shotgun bisulfite sequencing. It'll update itself every time you click a new thing. Under table, these are all of the different cell lines and tissues. So you should see the ones that we've chosen here, these ones, making sure we're choosing the CPG. So this is the replicate 1 and replicate 2. We only did the 1, so I'll choose that one. And unfortunately, you have to do this for each one. Um, there is a way to get them all on the same thing, but for the purpose of this tutorial, I'll just show you one so it doesn't get too overwhelming. Under output format here, I'm going to select the bed, otherwise known as browser extensible data, and I'm going to click get output for that. And this is what it'll look like. So you'll see that it'll show the genomic position. This here is the methylation percentage, so how strong the signal is of methylation at these specific coordinates. It also shows us things like whether it's on the plus or minus strand, and then the end coordinates. And then you can do the same thing for RNA-seq tracks to compare the expression levels on a more quantitative level. And so across these cell types, the HeLa S3 generally had higher promoter methylation and lower expression. So with the GM12878, we saw relatively lower promoter methylation and higher expression. This kind of fits with what we know about methylation repressing transcription, especially in promoter CPG islands. And TP53 is a major tumor suppressor gene, and the silencing of it through promoter methylation could contribute to uncontrolled growth and has been very relevant in cancer biology. So, observing these patterns visually helps us generate hypotheses about how epigenetic regulation contributes to cancer pathways. There are some limitations, so not all regulatory data sets exist for every tissue. Like you saw here, we didn't have the HeLa S3 cell line available to us. Um, Whole genome shotgun bisulfite sequencing coverage can be sparse, so the accuracy is not 100% confident. Um, and this is a visual hypothesis generation approach. It's not a full statistical analysis. So while this does provide some supportive data, um, some statistical analyses is required to confirm certain hypotheses predicted. But overall, UCSC allows us to do multiple things. It allows us to integrate multiple data types, as we saw here. It allows us to overlay regulatory features. We can visually interpret epigenetic relationships and compare tissues directly. And with that, I thank you for watching, and I hope this helped you see how to use UCSC for exploring epigenetic regulation. And feel free to follow along with the manual for more detail or replay any steps during your own analysis. Thank you.